Hello, my name is Eden McMillan Gallegos. I'm a senior studying biochemistry at Louisiana State University, and I conduct research on the biochemistry of inflammation. Today, I'm presenting my research on elucidating the open and active form of human 5-lipoxygenase. First, what is a lipoxygenase, or a LOX? A LOX is an enzyme that causes inflammation. They produce potent inflammatory molecules by oxygenating lipids. Human 5-LOX resides in leukocytes, or white blood cells. When signaled to begin the process of inflammation, 5-LOX moves to the nuclear membrane. Here, a phospholipase, PLA2, is also signaled to cleave arachidonic acid, which is a lipid derived from the membrane. 5-LOX then uses arachidonic acid as substrate. The 5-LOX reaction has two steps. The first product comes from a dioxygenation reaction that adds molecular oxygen to arachidonic acid to produce 5-HPEAT. This gives the name a 5 because the fifth carbon gets the oxygen. Next, 5-LOX forms an epoxide, producing leukotriene A4. LTA4 can be converted to other leukotrienes and go on to cause an inflammatory response itself. So that is the overall look at 5-LOX activity. When overactive, disease states such as coronary artery disease, asthma, and neurodegenerative diseases can arise. So this is one reason we are interested in the structure and function of 5-LOX. 5-LOX is a peripheral membrane protein, meaning it does not always reside nor act in aqueous solution. It is also intrinsically unstable. But because of the importance of this enzyme, our lab found ways to make it easier to study 5-LOX in vitro. In the past, we made a pseudo-wild-type version of 5-LOX, referred to here as stable 5-LOX. This is a soluble, stable version of the enzyme that allows us to study it in solutions. This model yielded a crystal structure for human 5-LOX. Here we see the active site iron surrounded by a mostly alpha helical region and two beta sheet regions on the sides. It is not emphasized here, but the structure showed a certain structural difference compared to other LOXes that stuck out to us. This difference in a certain alpha helix gave us a hypothesis about the potential for conformational flexibility of 5-LOX, which is the topic for today. Helix alpha 2 is drastically different in the 5-LOX structure compared to other LOXes. While 5-LOX shows a broken V-shaped helix, other LOX structures, such as 15-LOX2, show an elongated helix. Further crystallization experiments with 5-LOX by our lab revealed an elongated structure of helix alpha 2. This is pictured to the right. The global structure is the published structure for stable 5-LOX with helix alpha 2 emphasized in pink. And then in the box you see the broken helix overlaid with a gold helix with an alternate conformation for these residues. This alternate structure gave us the idea that 5-LOX may exist in an active state and a resting state, having access to the elongated helix that is seen in other LOXes when in an active state. Note that, in theory, an elongated helix alpha 2 would open a path for the 20-carbon substrate arachidonic acid to enter the active site. These considerations led us to ponder that an elongated helix may form during a conformational change, and we think that this could be functionally significant, potentially for regulation, membrane binding, and catalysis. Here's a movie showing what this conformational change might look like. Notice that more space is created by bulky residues moving away from the active site. These residues are hydrophobic, and in solution, their exposure on the surface would be unfavorable because aqueous solution is hydrophilic. We know 5-LOX is a peripheral membrane protein, so this movement of residues made us wonder if a conformational change could also assist in membrane binding. Shown here is a different angle of the structures we've been focusing on. Notice how two phenylalanines are pointing downward in the long helix. If this is where the membrane 5-LOX interface is, then these Phs could be involved in an event known as Phe insertion that is common for peripheral membrane proteins. This would make sense then for the Phs that are favorably buried within the protein while in the cytosol to move outward when given the chance to favorably interact with the membrane. 5-LOX also associates with a helper protein in the membrane known as 5-LOX activating protein, or FLAP. This change in conformation could have a role in the 5-LOX FLAP interaction as well. Because 5-LOX makes strong inflammatory molecules, it would make sense if a conformational change was also involved in catalytic regulation of the enzyme's activity. In addition to membrane and flap interactions, by creating a wider pathway into the active site, an elongated helix might allow a faster reaction. So to test this hypothesis that helix alpha 2 changes form, we use several biochemical techniques that allowed us to alter specific residues on helix alpha 2 and then evaluate the impact of the global properties of the enzyme as well as any alterations to the specific catalytic activity of the enzyme. In addition to knowing if an alternate form of the helix is available, we wanted to know the exact molecular determinants of this phenomenon. Knowing the determinants tells us more about 5-LOX activity in general as well, and it gives us a solid background for diving into the research that can treat disease states, one of which being structural-based drug design. For simplification, an open conformation refers to the state of 5-LOX with an elongated helix, and closed means that 5-LOX has a broken helix. We design mutants to favor either the open or the closed state. In green, you see two residues that we mutated to the neutral residue alanine in order to favor an open conformation. Glycine is a known helix breaker, so we didn't want that present. And the aspartate here forms a strong hydrogen bond in the broken helix, so we got rid of that as well. In the blue, you see two residues that we mutated to reinforce the favorability of a closed conformation. The hydrophobic Phes are already happily exhibiting hydrophobic interactions and also cation pi interactions within the protein. We capitalized on the positive arginine residues that are nearby but are not shown, and added negative aspartate residues to generate ionic locks, which is a strong type of bond. So now that we have our mutants, we want to see how they act in comparison to stable 5-LOX. We first notice oligomeric trends when purifying the enzyme by size. Size exclusion chromatography separates proteins according to size, and proteins can sometimes strongly associate with themselves, which increases their size based on SEC. This is shown on the left. We saw that stable 5-LOX could form monomers and dimers in a basic buffer, and it was fully converted to a monomer when the disulfide bond reducing agent TSEP and chelating agent EDTA were added. The closed mutant was a monomer regardless of the buffer, while the open mutant was a dimer regardless of the buffer. This first made us think, okay, if hydrophobic residues are exposed more often on the open mutants, such as what we see here circled in red, then what if hydrophobic interactions occurred between this interface of helix alpha 2 on the monomers? Are they looking for a binding partner when in the aqueous solution? This also makes sense for the closed model to not need a binding partner if the hydrophobics are rarely exposed. If these hypotheses ring true, this phenomenon could be important for understanding membrane and flap association as well, such as what's pictured on the bottom right. Our next global evaluation was conducted using limited proteolysis, a technique wherein the protein of interest is incubated with a protease, for us it was pepsin, at 37 degrees Celsius for 15 minutes. Pepsin cleaves at aromatic side chains, and I'd like to point out that based on the size of polypeptide fragments produced, we know that we did not remove a protease cleavage site by mutagenesis. When complete, we observed the relative change in SDS page band size by comparing the before and after proteolysis samples. So at the top, you see images of the raw data showing samples before and after 15 minutes of proteolysis. On the left, you see a bar graph depiction of this data from triplicate measurements. We first noticed that stable 5-LOX resisted proteolysis pretty well. There was not much of a change in band size. We also observed that the closed mutant resisted proteolysis cleavage as well, but the open mutant showed a significant increase in susceptibility to proteolysis. Only about 50% of the full-length protein band is remaining after 15 minutes. The data supports increased polypeptide flexibility of the open mutant. We interpret this to mean, yes, the open mutant has some new properties compared to stable 5-LOX, and the closed mutant mimics stable 5-LOX activity here. With that, we confidently moved on into a closer look at what is going on at the active site. We tested the activity by monitoring the production of the first product of 5-LOX's two-step reaction. This product gets hydrolyzed in solution to be a HEDE form rather than HPET, but the analysis is unaffected. HEDE absorbs at a specific wavelength, 235 nanometers, allowing us to observe an increase in absorbance over time. With the same concentration of enzyme and substrate for each variant shown in the graph and no enzyme activators present, we saw two interesting results. First, the pre-steady state lag phase, which is like the wind up before the constant or steady state activity occurs, was altered. The open mutant had virtually no lag phase, while the closed mutant closely mimicked the lag phase of stable 5-logs. Second, concerning the linear steady-state activity, the open mutant exhibited a sharper slope, which coincides with a larger initial velocity of Hd production. Our interpretation is that the data supports increased access to the catalytic site for the open mutant, which is consistent with our open 5-logs model. Furthermore, the previous observations at one substrate concentration was reflected across many concentrations of arachidonic acid. Shown here are saturation curves for the variants. Each dot reflects the initial velocity at a specific arachidonic acid concentration. Larger initial velocities for the open mutant and conserved initial velocities for the closed mutant compared to stable 5-logs were the result. The K-cat, or product turnover number, was increased by a factor of 4 for the open mutant. Similarly, the substrate affinity, reflected by the K-half, was around 4 times higher for the open mutant, as higher affinity is associated with a lower K-half. The data fit to sigmoidal curves, which is consistent with the idea of a conformational change associated with enzyme activity, considering that this enzyme activity held true for both monomers and dimers. Across the board, the open mutant appears to be acting in a manner that suggests increased access of substrate to the active site or increased product release, or both. The big picture here is that 5-logs and inflammation as a whole does have a role in many of the diseases people face, ranging from cancer to heart disease to asthma. If we can understand the underlying function of the structure of 5-logs, we are one step closer to understanding how disease states arise and how to design treatments that will be effective against overactive inflammatory responses. With only one FDA-approved 5-logs drug on the market, our research plays an important role in advancing the efforts of putting the brakes on inflammatory-related damage to the body. I'd like to give a special thanks to Drs. Marcia Newcomer and Nathaniel Gilbert for their mentorship and guidance throughout both my experiments and my project as a whole. Thank you also to my entire team for your support and collaboration on this exciting project. Finally, thank you for watching my presentation. I really hope you enjoyed it. I would be happy to answer questions or hear comments from you. Feel free to leave a comment below for me or my team of LOX researchers.